Hi, I'm Meredith Blackwell, and I am part of the group that is interested in the oral history project for Mycology. And we're here in Minneapolis, Minnesota at the Mycological Society of America meeting in Minneapolis. And we're here to, I'm Meredith Blackwell, this is Don Pfister, and this is Chase Myers. And we're going to interview Chase, who's a very recent PhD, and he's now on a postdoc. And I just want to say that I knew Chase as a person in David Longstreth's lab down the hall and around the corner. And I knew him there for a number of years. And not once did he take a mycology course or even come visit my lab. And then he goes off to Iowa State and becomes a mycologist. So, anyway, go on. Your turn. Where were you born? And
where was I born?
Yeah.
I was born in Baton Rouge, Louisiana, in the middle of Cajun country.
And are you Cajun?
75%, yes.
Okay. It's covered up by your name.
Yeah, that's right. It's a German name, but we got plenty of Cajun blood.
Yeah.
So you didn't get started by taking a mycology course from Meredith, but how did you get to graduate school in fungi?
Well, when I was working in Dave Longstreth's lab, I was convinced I was going to be a bacterial plant interactions person, because that was what we worked with. We worked with salt tolerance and these alligator weeds and Spartina grasses. And I was going to apply to Iowa State among the schools, and Dave suggested I talk to Meredith to get some advice on who I might work with, and she suggested I should reach out to a Tom Harrington who had a cool system there working with ambrosia fungi and ambrosia beetles. And so when I arrived at Iowa State, still not knowing exactly what I wanted to do, but pretty sure I was going to be a bacteriologist, I rotated through 3 labs. 2 of them, well, one bacteria heavy, one kind of a upper level meta communities lab, and then this fungus insect lab. And sure enough, it was the one I chose. So it was a good suggestion.
So Meredith really did get you to the place you wanted to be.
Yes.
I gave him a little bit of a
push.
push.
Yes. Yeah, that's right.
So tell us about
what
you did in Tom's lab.
What I did. In Tom's lab, I was lucky to take advantage of a really awesome system called the Ambrosia symbiosis, a diverse and severely understudied system.
What is ambrosia?
What is ambrosia?
Well, what is an ambrosia beetle?
Well, ambrosia is the food of the gods in Greek mythology. It's what they ate to maintain their immortality. But ambrosia beetles eat only one thing, and that's fungus. It's a fungus that they carry with them in pockets inside of their body. It's co-evolved symbionts that depend on the beetles to transfer them from tree to tree. And the beetles farm gardens of them inside the wood, and it's the only thing they eat. It's an incredible symbiosis.
And something that's always given me trouble is, how do you tell an ambrosia beetle from a bark beetle?
Well, if they're not in the bark. So it's a murky terminology, right? Because bark beetles are a huge and broad group, and ambrosia beetles have evolved 11 or more times from these bark beetles. And it's because they all did something magical, and they moved from living up in the bark and feeding on the phloem, the living tissue, and diving deep into the sapwood where the nutrients are scarce, but they brought fungi along with them to take advantage of that. So how do you tell if it's a bark beetle or an ambrosia beetle? Well, you can use context, see where their galleries are, where were they digging, and second, do they have mycanja?
Okay.
Now, bark beetles do have mycanja, but they only use fungi generally as a supplement to phloem tissue. Ambrosia beetles will only be eating the fungi they grow.
Okay.
And mycanja are those little pockets.
Yes. Little or big, they vary a lot in size
right.
and shape and where they're at.
And Chase has good photographs because I've borrowed one.
So what did you do for your PhD? What was your project?
Well, in short, my project was to sample a lot of different ambrosia beetles, especially ones whose ambrosia fungus symbionts hadn't been characterized well. Characterize those fungi and try to relate some pattern in evolutionary history to how those beetles got those symbionts, how did they domesticate them, and what explains the patterns we see today. And in the end, we were able to relate really interesting patterns in lineages of beetles, their unique types of mycanja, and fidelity with clades of symbiotic fungi, which included actually a number of novel new genera and species of ambrosia fungi, including, by the way, the genus Meredith.
He paid me back.
That's right, that's right.
And what's that fungus like?
Oh, it makes little Mardi Gras beads maybe.
Okay.
It makes maniloid hyphae that forms in these really long strings with these very small, very distinctive side branches of nutritious, yummy spores for the beetles to eat. But it's a unique morphology.
Yeah. And I should mention that long ago in my lab, one of my students, Stephen Kassar,
Yes.
was the one who really first found this and showed that the beetle fungi were, the asexual ones were also non-monophyletic.
That's right.
And
Yeah, so you guys had the first phylogenetic data that there was some cryptic diversity here.
scant, but
Yes,
but hard.
enough, and it was very
and
true.
Stephen was very much responsible for that because he had worked on it as an undergraduate, that project,
Yes,
with
that's-
somebody who's become a collaborator of yours.
Yes, with Richard Roper up at Alma College,
Right.
who was the first to notice that there were patterns in the types and complexity of Mycanja and ambrosia beetles and the clades of symbionts they carry in those Mycanja. So
Yeah.
he was actually instrumental in coming up with the impetus for this project. Yeah.
Yeah.
So you're at Cornell now.
Yes.
What are you doing there?
I'm doing something a little similar to insect-fungus symbiosis, except not. I'm doing endobacterial symbiosis or robuscular mycorrhizal fungi. So I've gone from dealing with insects that carry fungi and eat them to obligate symbionts of plant roots who have obligate endobacterial symbionts.
So you're back to bacteria.
Yeah, that's right. I ended up there anyway, after all.
So are these basidiomycetes or are they
No, these are
zygomycetes?
well, under the current terminology, they are
Mucoro.
subphylum Glomeromycotina
Okay,
of the
okay.
phylum Mucoromycota.
Okay.
Yeah.
That's right.
Yeah, yeah, and I know some of the rhizopus that have some effect too. They live
That's right.
within.
That's right. So yeah, my main focus is on the Glomeromycotina and their endobacteria, but we have side projects on rhizopus of the Mucoromycotina, who have their own very interesting endosymbionts.
And that could maybe get you back to Louisiana because of the rice, because I
Right.
know some of them
Yes.
are in rice.
Yeah, and that would be full circle to bacteria and geography.
LSU needs a mycologist or somebody who studies symbiosis in the biology department.
Yes.
So we'll have to
Go
make sure they
Tigers
keep
and
you
go
in
fungi.
mind.
We're publicizing it right now.
Yeah, yeah. We
Sure.
just have to
Oh.
make them let us have a job to advertise.
Okay, great. Yeah, LSU needs strong mycology.
Yeah,
It's got
yeah.
a long history of it there.
Yeah,
Yeah.
yeah. So, okay, so you're going to do the postdoc, and you're working with Paula.
Oh, I'm working with Teresa Pawlowska
Teresa,
right
okay,
now. Yes.
okay, yeah.
But Paula did a lot of instrumental early work in the endobacterial symbiosis over at Torino, Italy.
Okay. Yeah, and then Greg at Michigan
Yes,
State
Greg
is working on
Benito.
it too. Yeah.
Yes.
Yeah.
Yeah, so Greg's work has been focused on the Mortierella mycotina, another understudied group of these early diverging fungi. And they found, he and his student Alessandro De Ciro found a surprising number of diverse endobacteria in these fungi. So,
Oh. So what do you plan to do when you're done? Do you have any idea? Depends on
what
what's
time scale are we talking
available.
about?
Or
Yes. Everyone can write up their dream job. So should I tell you my dream
yeah,
position?
tell us
Yeah,
your
yeah.
dream.
I have a real passion for teaching, and so I would like to end up in a position where I can teach a lot, but that I can innovate in the teaching, and I can continue to iterate, use new active learning techniques, but I'd like to connect that with research if I can, basic research. And so if I was writing the job description, it would be a kind of balanced teaching and research position where I have control of a lot of very excited and passionate students who are willing to go and sample fungi out from the environment and characterize it. And I try to funnel some of that into basic diversity explorations. Perhaps I would sample from among that diversity and find novel taxa and start to describe new systems. Research-wise, I continue to be interested in how fungi interact with other organisms. At this point, whether that be insect or bacteria, I want to keep the theme of fungal interaction with other organisms and how those interactions evolved over evolutionary time.
Okay. Interesting.
Yeah.
But I want to keep the teaching thread in there.
Yeah, that sounds wonderful.
Yeah, yeah.
Just be careful about thinking that you can control students.
Yeah, that's fair.
That's sometimes a problem, but I think you could do it.
Well,
You
if
have
you
to
can-
excite them too.
Sure.
Yeah.
If
You don't
can-
want to.
Sure.
They're not going to come in excited.
Sure, but if you can't control them, sometimes you can nudge them in the right direction
That's
and say,
right.
why don't you try
That's
out
right.
fungi?
Exactly.
Yeah.
Exactly.
Yeah.
So, I think we're getting cut off here.
Okay.
Okay.
So
Good.
anyway,
But thank you.
Yeah, of
Thank
course.
you
It's been
for
great to
taking
talk to
time.
you.
Yeah. Did I mention that it was Tom Harrington and that I did the PhD with? I
Yeah,
did.
yeah,
Okay.
yeah,
Good.
yeah,
Good.
yeah.
Good.
Yeah.
Good.
No, no, no.
Want to make sure I included that in the
I think
story.
you said it 2 times.
Great. I
Yeah.
don't think there's anywhere else I could have worked with this system. So
Yeah.
And he gave you a lot of freedom, I'm sure.
absolutely.
Yeah.
Yeah. Which is great.
Okay.
Cool.
Okay.